And just to set this up, this is specifically looking at regulatory trials and LSTs. And it's also a way for us to investigate who's causing all the complexity in clinical trials. Whose fault is it? And so we have a couple of candidates here, potentially. And I'll just start it off by talking about experiences I have in my world. I won't name a study, but we did discuss in the planning committee a study that we're currently running that's for a single product to be approved on the medical device side, which has approximately 2,800 fields of data, 7 different core laboratories. It takes the average study coordinator 32 hours to do one patient and probably another 30 to 60 hours to do the monitoring on top of that. The cost per patient is in the hundreds of thousands of dollars, and it's for the approval of one product. And so in my own kind of investigation, I talked to the business unit and wanted to know, how did this study get to be so big, expecting them to say, oh, it was the Food and Drug Administration, they made all this stuff happen. It actually didn't go that way. It turns out that a lot of the fields came from the company, from ourselves, because we were basically trying to cover all the bases. A lot came from the principal investigator, who wanted to pad their potential data sources for papers to be written for the next 10 years. And then that principal investigator also had connections to core laboratories that they wanted to actually put on top of that. So it isn't an easy answer as to why this happened. So that leads me to a potential Mike Lauer metaphor here. In 1962, the NFL championship was won by the Green Bay Packers over the New York Giants. The score was 16-7. The quarterback for the Green Bay Packers was Bart Starr, who had 3 interceptions compared to the New York Giants quarterback who had 1 interception. A variety of other facts about the game, which actually went in favor of the New York Giants, but Green Bay Packers won. At the end, Vince Lombardi quipped that statistics are for losers. And the notion there is that if you win, you don't need the statistics. If you lose, you quote a lot of statistics. So what does it have to do with LSTs? I'll get there. The idea is that I think there might be a lot of pressure on both the regulatory agencies as well as companies, especially regulatory path studies, to put a lot of data into studies just in case things don't go right. And my guess is, and I'd love to hear Rachel's comments on this during her presentation, that when you actually meet your endpoint and your hypothesis is rejected, likely you don't need to have all that extraneous data to explain what happened. And so a lot of the notion is the issues of what happens when you don't meet your endpoint. And it'd be interesting to see when that happens, has that been a successful pathway? And also strategy, that if you don't meet your primary endpoint, do those mounds of extra data actually help you get a product approved? And is it a myth? And so there are a lot of issues I think we can explore about why the data are there. And so we have 3 speakers here. Let me just quickly introduce them. Ken Getz will start, and I think he's online. And I'll just get a little background about Ken. Ken's the director of the sponsored research programs at Tufts Center, and he's also an assistant professor there. He studies R&D management practices and pharmaceutical and biotechnology company operating models. And it basically looks at the interactions between global investigational sites, outsourcing and study volunteer practices, trends and policies in doing large-scale studies. He's also chairman of CISCRIPT, which is the nonprofit organization that was founded to educate and raise public awareness of clinical research enterprises. And he's also founder and owner of CenterWatch, which many of us subscribe to, which is leading publisher in the clinical trials industry. Chris Granger is a cardiologist, professor of medicine at Duke University, and director of the cardiac care unit at Duke University. Chris is involved in a lot of journals. He's an editor of the American Heart Journal. He's on the board of editors for the Journal of American College of Cardiology. And he's currently the cardiac section author for the current medical diagnosis and treatment. He serves on a variety of publication oversight committees for the American Heart Association, and is the chairman of advisory working committee of the American Heart Association Mission Lifeline Program. His primary interest is in methodologies and conduct of randomized controlled clinical trials, so he's perfectly suited for this discussion. And Rachel Sherman is Associate Director for Medical Policy, Center for Drug Evaluation Research at the U.S. Food and Drug Administration. She's responsible for developing, implementing, and coordinating medical policy programs and strategy initiatives, including regulation of prescription drug promotion and advertising through the Center's Division of Drug Marketing, Advertising and Communications. She provides leadership and scientific guidance and advice in clinical trial implementation, facilitates the development and implementation of the agency policy related to human subject protection and good clinical practices. So she's perfect for this discussion going forward about who's at fault for these big clinical studies and what we're going to do to make them smaller. So with that, let me introduce Ken and see if we can make the connection for his talk.
Great. Thank you so much, Rick. I just want to do a check before I go into my presentation. I hope all of you can hear me and you can see my slides.
Yes, we can.
Wonderful. I just want to say thank you very much for your introduction, and to all of you, I'm delighted to be joining you today, and just so sorry that I could not be there in person. What I'd like to do is share with you about 10 years of metrics and statistics. I'm not sure what that says about me. Based on your comment a moment ago, Rick, but we've been studying protocol design complexity and its impact on performance for over 10 years. And what I'd like to do is share with you some of the characteristics about our protocols, how they've changed over time, and we've been able to quantify that. And then looking specifically at how those changes are impacting the performance of our clinical trials. Rick, you did such a nice job of setting the stage, and I'd like to share with you a representative Phase 2B3 registration trial today. The typical study has an average of 13 endpoints, 1 primary, 5 key secondary, and the remainder are essentially tertiary and exploratory endpoints, some what we would call supplemental secondary endpoints. But that has been the area where we've seen the most growth in terms of the total number of endpoints supported by protocol procedures. It's been in that area of exploratory and tertiary activity. And in a moment, I'll talk about our thoughts on why we see so much endpoint creep, if you will. The typical protocol has nearly 170 procedures. This is an average across all therapeutic areas, and half of those procedures are supporting the primary and key secondary endpoints. The typical protocol has an average of 35 eligibility criteria and nearly 170 case report form pages. And in a moment, I'll show you how that has, these two areas have changed over time. And the typical protocol requires the study volunteer to make 11 visits over an average of 175 days of participation in our trials. All of the data that I'm now going to share with you, it has been published in peer review articles, some of which I've shared with you with your material. And if you'd like to see the details of these studies, you can also send me an email, and I'd be happy to send you the PDF articles, or our librarian will help to do that. This next chart really summarizes the 10 years of research. We've broken our measures of complexity into 3 observation periods, and there are a wide variety of measures that we can look at. We can look at the number of unique procedures that are conducted to support a protocol. We can look at the total number of times or frequency of each unique procedure and aggregate them. We've created a new measure called site work burden, where we look at the typical individual who performs a procedure and essentially their value, their time value, and the amount of time it takes to actually administer that procedure. And then you see some other measures as well. In every case, we've seen a dramatic increase over these 3 observation periods. And what's driving that? Well, many, many factors, and I think, Rick, you articulated some of them. We've seen a real shift in our pipelines from a focus on acute illnesses to chronic illnesses, for which endpoints are far more difficult to measure. Many of our studies today are collecting more genetic material and biomarker data. Many of our studies now involve a combination treatment or a diagnostic, and so those have added complexity and additional procedures. We also have seen a shift in our scientific strategies. In our Phase 1 studies, for example, we're collecting a lot more safety data, and many would argue that that's in part due to pressure from regulatory agencies. In our Phase 2 programs, we see the tendency to collect more data in order to determine whether a project should be terminated early before moving on into later stage and more expensive larger scale phase 3 programs. In phase 3, we also see a dramatic increase in all of these different measures of complexity, and that is in part due to additional data collected in anticipation of a regulatory request, and there are other reasons which I'll get into in a moment. And our phase 4 studies have shifted from more observational in nature to more robust controlled clinical trials. So this essentially articulates the scientific complexity of our studies. We can also look at the operating complexity, which also is quite pronounced today. Here we've just pulled a number of measures that we monitor routinely, and I'll just highlight a few. Our phase 3 programs are typically conducted in an average of 34 countries. We've seen some pivotal trials that are conducted in over 80 countries. And of course, when we conduct them in such a broad dispersion of countries, we are now forced to interact with multiple health authorities, different regulatory agencies. The challenge of distributing clinical supplies or collecting lab data in ever more remote regions adds a lot of complexity as well. That last data point, the last row, is quite interesting. We're collecting nearly 1 million data points for the typical phase 3 study today, just again to give you a flavor for the level of complexity that we can characterize and quantify. Well, in our research, we have tied the complexity of our protocols to performance measures, and I've cherry-picked a few here that I wanted to highlight. We know, for example, that far more complex trials actually have far worse recruitment and retention rates. It's much more difficult to keep patients in our studies. Our cycle times increase dramatically, but with an important point to make here, we only see a modest increase when we look at the time from when the protocol is deemed ready to getting the first patient in the study. It's really the downstream impact of a complex and demanding protocol where we see the real effect of complexity on extending cycle time. We also see a very strong positive correlation between the complexity of a trial and the number of amendments. And for those of you who've ever been involved in implementing amendments, you know how disruptive and how costly these can be. Our research has also focused on trying to isolate opportunities to streamline or improve protocol design practice. In our studies on protocol amendments, we've found some very interesting insights. We've learned, for example, that the majority of our protocols have at least one amendment, and the table on the left shows you the average number of amendments per protocol by phase, as well as the number of changes that each amendment triggers. What's interesting is we've learned that nearly half of all amendments are occurring before the first patient receives the first dose, and many have concluded that that indicates that companies are often hastily putting the protocol into the clinic without spending enough time assessing the feasibility of conducting or performing that protocol. In all of our studies, we ask the sponsor companies to classify their amendments or their procedures. In this case, we asked them to determine whether an amendment they felt was somewhat or completely avoidable or unavoidable, and we found that nearly 40% of all amendments are deemed avoidable. Many of them are the result of protocol authoring policies that are not appropriate or relevant for the current project, that oftentimes the protocol may include typographical errors or a simple design flaw that wasn't caught, and as a result, the protocol had to be pulled from the clinic, reviewed and approved, and submitted back to the Ethical Review Committee for review and approval, and the patients reconsented. The important point is that a protocol amendment is not a problem. It's a solution to an underlying design problem. And we've been able to quantify the impact of protocol amendments on cycle time and cost. The typical amendment adding roughly 2 months of additional time to a study and costing about $500,000 in direct costs. So that doesn't include any of the staff time to manage and implement the amendment. The last area I want to touch on is a recent study that we've just completed, and this will be published in the December issue of the American Journal of Therapeutics, where we really asked the question, is all of this complexity necessary? Is all of it meaningful? For this study, we had each sponsor company look at their specific protocols and categorize each of the procedures along the following lines. And this was all according to the clinical study report or the statistical analysis plan. If the procedure was tied to a primary or key secondary endpoint, it was considered core. If it was a procedure that was primarily associated with a GCP/ICH compliance, it fell into the required category. If it was a procedure that was typically performed whenever the patient presented at the center or clinic, it was considered a standard procedure, and many could argue that a lot of the baseline activity that we conduct at every patient visit is not essential or necessary, but we've now included that in the standard category. And the last area we call non-core or less essential procedures, and these are those that support the supplemental secondary as well as tertiary and exploratory endpoints. And here's our findings. We found that roughly 1 out of every 5 procedures that are performed are actually non-core in nature. And again, these are classified by the sponsor, the developer or designer of each of the protocols that they contributed. Now, why would we see such a high proportion of our procedures falling into that group? Well, there are many reasons. Companies often are looking to collect additional data to provide context to guide their interpretation of their observations. We also know that companies are looking to collect additional data for insights into a particular mechanism of action or a new insight into a potential new area of development. But we also know, as Rick had suggested at the outset, that there are also a number of efforts to mitigate potential risk, the risk of a request for additional data by a regulatory agency, anticipated resistance by purchasers and payers once a drug is approved and in the market. And we also know that there are certain habits that translate into additional procedures. For example, companies will take a protocol design from an earlier phase project and then simply apply and build on that design for a later stage study without often asking the hard question, are all of these procedures necessary? Again, it's so important to keep in mind that as we add more procedures, we challenge the performance and adversely impact the performance of the trial. We also looked at the direct cost to perform each of these procedures, and roughly 18 cents on every dollar that's spent on our clinical trials is going to fund our non-core or less essential procedures. When I first presented these findings, a reporter published an article with the title, "Tough Center Concludes That Clinical Trials Are a Waste of Money." So somehow they really missed the point here, which is again just to suggest that there are areas where further scrutiny would assist us in simplifying our protocol designs. And the last area I want to touch on are just some of the differences by phase. And here you see that our non-core procedures in our phase 3 studies represent a substantial portion of the budget, that 18% figure, but approaching $2 million per study budget. If you actually look at the total number of active phase 2B, phase 3 studies that are conducted around the world to support our registration studies, we're looking at roughly $4 to $6 billion a year spent on these non-core or less essential procedures. So I'll end there. And Rick, I wasn't sure if we'll hold for questions until the very end, how you'd like to handle that.
Yeah, if you could hang on, we'd like to just do the questions at the very end. But thank you very much for your presentation.
Absolutely.
And now we'll move on to Chris. We'll talk about simplifying trials.
Great, thanks, Rick, and thanks to Ken for providing a very nice backdrop for my my presentation. And I'll start out with these statements that we can define trial quality, that large simple trials are burdened with complexity, as Ken has nicely pointed out, some of which does not improve reliability or quality, that there are examples of trials that are much more simple and fit for purpose. And the trials could be either radically simplified for answering some questions or incrementally simplified in most circumstances, and that cost reductions resulting from sensible simplification and focus on efficiency can be quantified, and that's what I'll really focus on. Here's Rob Califf. This randomized double-blind trial involving over 20,000 patients was conducted over a 10-year period. Unfortunately, we've forgotten why. So the first thing for trial quality is, as several people have pointed out, is that we have a good question that's clinically, that's important, and will change practice and will improve outcomes. So assuming that's the case, then here are the other elements of quality. And the reason that I'm just reminding ourselves of these is because I think these are the things where we really should be focused on in terms of the processes that we're using in trials that oftentimes now I think we're not looking at the mission closely enough, that we have an adequate number of events to answer the question with confidence and precision, that it's done in a practice setting which makes the results generalizable with proper randomization, with reasonably complete follow-up and ascertainment, especially of the primary outcome, with appropriate safety assessment, including not based on individual reports, but based on aggregate information, with a plan for continuous quality improvement in the context of that trial, rather than just a retrospective assessment at the end of the trial, with safeguards against bias, including blinding when appropriate, and with protection of rights of research patients. And some of the data I'm going to show you is now actually 5 years old, and it's worthwhile, as others have pointed out, that even in the last couple of years there's been substantial increase in complexity. And I'll draw from work that Eric Eisenstein led that came out of the first of 3 meetings that many of you have been at that were really organized by Salim Youssef with the collaboration of the Oxford and Duke groups, this one from 2007 when Eric led this review of opportunities to reduce cost in large trials. And these are pharmaceutical trials, phase 3 trials, and took some assumptions and then ran 3 different scenarios: a full cost, a model, a streamlined model, and then a more radically streamlined model. And this was the set of assumptions. This is a chronic disease trial of 20,000 patients, 1,000 sites, 2-year duration, 60 case report form pages, 24 site visits, and a 10,000 per patient study award. And in terms of where these costs go, a substantial portion goes to direct labor, and then then the non-labor costs, a large portion of that to site payments, which goes primarily to funding study coordinators, about half of the total cost, these site payments, and then some additional expenses for this total of about $400 million. And then in terms of the simulations, the components that were varied included the duration of the trial, the size of the trial with respect to case report form length and number of sites, not number of patients, and then some of the operational issues, and then a combined model. And no surprise to this group will be simply pointing out that we do have this challenge in identifying sites that are high quality and productive in trials, and it tends to be a minority of our sites that produce the majority of enrollment. These data from Lisa Berdan and our experience at the DCRI, where the top 25% of sites in phase 3 trials have enrolled about 2/3 of the patients, the top 10% about 40% of the patients, and then a large number of sites are unproductive. The challenge is identifying who these sites are, because they're not always, although probably the best predictor is how sites did in recent similar trials. Even that is not a perfect predictor. And then it costs a lot to start up each site, so that there's a lot of preventable expense in bringing up sites that are not going to be productive. So I think some of these are interesting in terms of of where it looks like the major cost savings lied in this particular set of simulations. And with respect to reducing time of planning and time of enrollment, relatively modest costs there, only about 1 to 2% reduction in total cost of the trial with those particular parameters. More opportunity in decreasing the number of variables collected, the case report form length, and especially the number of sites. And this is something that the Oxford group is really focused on, is that if one can spend more time identifying fewer productive sites, that's a major opportunity for reducing cost, almost 10% reduction in cost with a modest reduction in the total number of sites. And then electronic data capture, the assumptions here including effects on the coordination centrally of the trial and at the sites themselves, including a shorter closeout time, elimination of a lot of work in queries and data cleanup and medical coding, and then decreased time and energy at the sites as well. And then some site management assumptions, these being substantially streamlining some of the procedures in trials with a reduction in visits, actual physical visits to sites from 50% down to 10% of sites, and fewer visits, and essentially no closeout visits, and much less source data verification, down from 100 to 10%, focused on key variables and some random auditing. And then these had the biggest impact on reducing costs, where the use of electronic data capture reducing costs by about 10%, and streamlining site management decreased the cost by 21%. And then if one then took these in combination, one found about a 1/3 reduction in total cost from these assumptions. And then if one assumed that this would result in substantially less personnel time at the site, and therefore a lower site payment, here a 50% reduction in site payment, then this would result in a dramatic about 60% reduction in total cost. And then finally, the very streamlined approach, limiting to a very small number of very high volume sites, eliminating on-site evaluation, source data verification, very focused case report forms, and then a dramatically reduced site payment resulted in a much more dramatic, about tenfold reduction in cost. And then this is simply a display of the effects of the streamlined and the transformational streamlined approach in terms of the total cost for this type of a trial. And let me build on the discussion from the UK experience with this one example. Now, this is not of a new drug or device. This is a trial led by Stefan James and the Uppsala group in Sweden, built on top of the Swedish acute MI registry, that took patients with acute MI and randomly assigned them to thrombus aspiration with primary PCI or primary PCI alone, and used the registry really to do almost all of the trial procedures, including enrollment, randomization, limited additional variables that were collected, and follow-up. And here, built into the registry electronic interface itself, was the identification of eligible patients and the registration of those patients, and collection of whether or not the patient had provided verbal informed consent, and the inclusion-exclusion criteria were met. And then this is a registry that's really very advanced and incorporated into care in Sweden, such that all hospitals in Sweden participate in this registry. And then you see, of all primary PCIs performed over the last now 2 years or so, or at least a year, that the majority of patients were randomized into this trial. And the total incremental cost, now this isn't the true cost because the trial was, most of the cost of the trial is already incorporated into the registry and patient care. But the incremental cost of these simple procedures in the context of the trial only about $50 per patient. So for a total incremental cost of $300,000, now the trial has increased the sample size to 6,000 to 7,000 and is now completing enrollment. So just another example that it is possible to do this in a highly organized healthcare system, looking at a simple procedure with clinical equipoise and with physicians that are enthusiastic. These interventional cardiologists really wanted to have this question answered, so they've all been very eager and supportive and enthusiastic in enrolling their patients. So then the question that we've, I think, all been asking is, if one can save all this money, why hasn't it already been done? I think these are some of the reasons: the risk aversion issues that that Rachel and Bob Temple and others are very articulate in the opportunities to simplify things, but the reviewers and auditors don't always share exactly the same perspective. That in terms of missing something, maybe sometimes we would rather collect unnecessary things than be frustrated by missing something important. That we have this research industrial complex that exists, I think, in each of our institutions, in academic research groups, in regulatory departments, in large pharmaceutical and device companies, and certainly the CRO industry, which has absolutely no interest in simplifying things. In fact, their business model and margins are created by the complexity that has been established, and they have substantial, therefore, monetary stake in maintaining the status quo. And then the lack of international harmonization does, in these international programs, oftentimes force one to divert to the most complicated common denominator. So then each trial is different. This is part of our challenge, that there's no simple answers, there's no universal answer, but that there are these opportunities that vary amongst sponsors and treatment studies and stage of development to substantially reduce costs of these large-scale clinical trials. And I think at least oftentimes not compromising quality, and in fact sometimes even enhancing quality, and that we can look at this in an incremental approach and in a transformative approach. And finally, and again, to Ken's credit for being a world leader in this, that we really need more research in terms of the processes that we use in clinical research. For a group that that's so dedicated to the foundation of evidence-based medicine, it's surprising how little we've actually studied the processes that we use in these trials and what additional value we get from them. Thanks for your attention.
Thanks, Chris. Rachel?
Good afternoon. On behalf of the agency, I thank Rick and Dave and the forum for including us. It's a real pleasure to be at a meeting where we're not being blamed for everything from global warming to I don't know, striking the least. A couple of disclaimers. This is going to be a drug-centric presentation. I know drugs best, but my colleagues Doug Pratt and Graham Zuckerman are here representing the other centers. So if I say something terrible, they'll scream. And I think I'll start off by, in response a little bit to what I usually say to sponsors, which is talk to us early and often, and if we say something that doesn't make sense, ask us again. That is the single most common mistake I think that industry makes when approaching us. And obviously, I will speak primarily from perspective of the agency and regulated industry. I think Deborah underestimated the number of trials that are not going to be productive in terms of what as a reviewer and a team leader, I used to call it safe but stupid research. Now I'm a little older and wiser, and I think as per comments made yesterday, in fact, it's unethical if it's not going to produce anything useful. If we think of the resources that are available in this country and around the world to address the evidence gap we all know exists. I think about it as a little tube of blood from a pediatric patient instead of from an adult patient, or we have a credit card with a limit. We can't go on spending like this or using all that blood. We're not taking the blood from a 70-kilo man. So if we adopted that attitude, we wouldn't be throwing on all these extra fields. And I think publications being what it is, we have undertaken in my office a look at late-stage failures, which I think we're all. And certain things, it's really shameful. No one should go into phase 3 not knowing the dose, for example. But in answer to your question, Rick, we've never seen a program, at least I've never seen a program, and we certainly didn't find one that was saved by either the statisticians when they fail on the primary endpoint, call on the statisticians, saved by that extra data. If you make it on the primary endpoint, the pre-specified primary endpoint, you're in good shape. All right, so what did I... And I'd also like to thank Patrick Archdeacon from my office who filled in for me when I was out for a little bit and helped put together some thoughts for what we could say today that might be useful. And in thinking about it, actually, it's a topic very near and dear to our heart. When we think about clinical trial design, we think about data quality. It really all comes back to data quality. It's a very old concept. It's a very modern concept. And Rick, I don't know if you picked 62 because I picked 62 or because it was a year, but 62 is a very, very important year for us because, let's see if I can do this. 1962 was the year that the of the Kefauver amendments where the efficacy standard, which I can always get a little choked up over, was passed, and I will read it because I think it's really important to know that not only can very important legislation be passed, but it can pass the test of time. It can still be useful 50 years later, and it can still withstand the every 5-year assault, or sometimes more frequently, that it undergoes. So our statute is written sort of backwards, and it says when the secretary should deny it, but it refers to substantial evidence and says substantial evidence means evidence consisting of adequate and well-controlled investigations, including clinical investigations, I'm getting choked up, by experts qualified by scientific training and experience to evaluate the effectiveness of the drug involved, on the basis of which it can fairly and responsibly be concluded by such experts that the drug will have the effect it purports or is represented to have under the conditions of use prescribed, recommended, or suggested in the labeling and proposed labeling thereof. That has all the elements we all need to design and conduct sensible clinical trials, in my opinion. So what are our obligations? It does not prescribe a particular, and this is our law, by the way, so it's enforceable. It does not prescribe a particular trial design. What it says, it has to produce evidence, has to be appropriately designed, and there are words elsewhere, all tests applicable, appropriately designed, and then produces useful information, and then we have to communicate that information. So those are our responsibilities by statute, by law. And if anyone hasn't read it, there's a very interesting commentary recently in New England Journal. Actually, what Kefauver was after was a comparative efficacy standard. This was back in '62. As usual legislation, he didn't get it, but it's just interesting to note that that's really one of the things that was in play. So what about our regulations? This is, again, if we're talking about if we're nervous that FDA is going to come and inspect, as I know Rob always reminds me we might send a fish inspector to do, what is it that we, or you're going to come and meet with a reviewer, what is it that we can require? Well, we do, and do we ever, remember this conference did talk about large, simple trials, or was entitled that, although I think what we're really talking about is streamlined, sensible trials with one-page informative consent forms, I would add. So what else do we say? Well, the regulations do talk about what an adequate and well-controlled trial is, and I think people are pretty familiar with that. But once again, in addition to listing the types of controls such as placebo, dose response, et cetera, what's embodied in the regulations is that the study uses a design that permits a valid comparison with a control to provide a quantitative assessment of the drug effect. So again, it's an emphasis on the quality of the data, not a specific design, but the quality of the data. And that gets back to the point that's been mentioned over and over again. Ask a good question. If we ask a good question, we may get a good answer. And it talks about minimizing bias. Where else do we talk about trial design in our regulations? Well, one of the only other places, interestingly, is the clinical hold. So when can we actually stop a study? Well, I'm going to go to the second bullet first. In phase 2 or phase 3, we have the authority to stop a study if it's clearly deficient in design to meet its stated objectives. That's the only place we can actually stop what I consider, what I used to term safe but stupid research. We can, interestingly, put on hold or stop a trial that is not adequate and well controlled, but only under certain circumstances. And that primarily those circumstances are if it's going to interfere with adequate and well controlled trials that are going to get us the kind of data that we need. Other than that, we do not have the regulatory authority to put it on hold for simply that reason, that it's not adequate and well controlled. And obviously the most important thing, and I think a point that was slightly missed in the morning, is the first thing is if there's presents unreasonable risk. And we talked a little bit this morning about I forget the precise expression, but the physician friend and the physician researcher. Well, in our regulations, and I think any clinical trialist would agree with this, the patient always comes first. If there's any question of risk to the patient, that trumps. So, for example, I mean, none of us ever want to unblind a study. But if a patient gets sick and we need to know whether or not they were on the test article, they are unblinded. There's no question. So I think it's very clear what the hierarchy is there. The patient comes first before the, if you will, integrity of the study. So those are the places in our regulations, again, enforceable, the regulations, where we discuss trial design. Oh, I'm on yellow already. Okay, so harmonization always a problem. ICH has some general, I think more, if you will, textbook-y discussions of trial design, and then guidances. And some of these have been hinted at, and these express really the agency's current thinking. I have 1 minute and 11 seconds. I'm doing so badly. In terms of streamlining, we've been pushing since, really since that first Sensible Guidelines meeting back in whenever that was, and working with Duke in terms of clinical trial modernization and streamlining clinical trials, because we know that the credit card does have a limit on it. So AE Light is what we affectionately call the guidance talking about, if you will, streamlining data collection in late-stage clinical trials. If we already know a lot about the safety profile, don't spend time and energy and resources and taking patients' blood and other specimens to reprove what we already know. Look for what we don't know. Similarly, oversight of clinical investigations, a risk-based approach to monitoring. Again, because Rob kept yelling at me about the investigators coming to a site. The notion is quality by design. Collect only the data you need and then only monitor what you need to know. And best of all, up front, start with a discussion with the agency so that when we all agree up front what we're going to look at, what we're going to collect, and then what we're going to monitor against. I didn't mention here the post-marketing safety reporting rule, which has been alluded to. We did issue new regulations. We do not want to see 15-day reports on everything. It was warmly welcomed by some in the industry who found it very efficient and cost-saving, not warmly welcomed by others when we actually had to delay implementation for a year, which surprised us to no end anyway. In any event, the agency does not emphasize a particular type of trial. We emphasize high data quality, not quantity. Once the trial is completed, we will look at the data. We will look at its generalizability. We will communicate that, and we think this is the most sensible approach. So I think what we'd be very interested in hearing, because sometimes our silence is deafening, is that if there are ways we are not promoting the use of more efficient designs, we would like to hear that. If there are ways that we can advance the conversation and advance the field, we'd like to hear that. Thank you.